My name is Clodagh Walsh and I'm a PhD researcher supported by H&H Group and based with the H&H Research team in Moore Park, Ireland. I will use this interview today as an opportunity to present findings from the HMO Biotic research platform and give insights into the human milk oligosaccharide sharing strategies employed by a consortium of infant-derived Bifidobacterium species. In advance of presenting findings from this work, I'll provide some background on what is known about Bifidobacterial species and the significance of their interactions with human milk oligosaccharides. Bifidobacteria are known to confer health benefits and are dominant in the gut microbiota of breastfed infants. Their efficacy and safety has been shown routinely in clinical settings, and they are therefore ideal candidates as probiotics. A key adaptation facilitating the dominance of Bifidobacteria in the infant gut is their ability to consume breast milk sugars known as human milk oligosaccharides, or HMO for short. It is hypothesized that species and strain level variances in the ability of strains to utilize HMO may impact their fitness and performance in the gut. Therefore, the development of HMO Biotic products should consider the selective consumption patterns of Bifidobacterial species and the cross-feeding relationships that exist during utilization of HMO. Therefore, the objectives of this work were to enhance our understanding of HMO microbe and microbe-microbe interactions to optimize the development of HMO-containing products. In order to characterize Bifidobacterial interactions with human milk oligosaccharides, we first isolated HMO from breast milk. Before cultivating strains on select strains on HMO, the strains selected include B. bifidum R71, B. breve M16V, B. infantis R33, and B. infantis M63, as well as a co-culture of the 4 Bifidobacterium strains together. Phenotype profiling was performed, followed by unraveling of interactions via omic technologies, including genomics, proteomics, and metabolomics, in order to provide a mechanistic definition of HMO Biotic benefits. Here we provide a summary of the growth of the strains with isolated HMO as sole carbon source. Interestingly, significant differences were observed between the 2 Bif infantis strains, with higher maximum growth and faster growth rate observed during co-cultivation of the 4 strains together. Most notably, while M16V demonstrated limited growth in monoculture, when qPCR analysis was performed on the co-culture, it was found that M16V was a dominant species within this co-culture. The variations in Bifidobacterial growth were similarly reflected in the rate of oligosaccharide consumption. Here we show the concentrations of the major HMO sugars prior to initiation of Bifidobacterial growth in comparison to the levels of oligosaccharide at the end of fermentation following growth of R71, R33, M16V, M63, and the 4 Bif co-culture. Again, notable differences were observed between the 2 Bif infantis strains, and the highest levels of HMO consumption were observed during co-cultivation of the 4 strains. Limited HMO consumption was observed in M16V, while high consumption was observed in R71. Notable increases in 3FL and HMO degradants such as sialic acid were also observed. Genomic mining and profiling was performed in order to provide mechanistic insights behind the growth phenotypes observed. In summary, we found that the significant differences between the 2 Bif infantis strains were due to R33 lacking the transporter for LNT and LNNT. The release of HMO degradants by R71 was due to the extracellular glycosyl hydrolases harbored by R71. By hydrolyzing HMO externally, R71 released HMO degradants such as sialic acid and fucose, which could be made available to other microbial species within the community. While B. breve M16V lacked most enzymes required for consumption of complex HMO, the enzymes for metabolism of HMO degradants were found to be highly abundant during co-cultivation, especially with R71. Here is a short animation which I will use to demonstrate what happens between these Bifidobacteria in a shared feeding environment. Some strains, such as B. infantis M63, gobble up everything available to them and don't particularly like to share, while other Bifs, such as M16V, are a little more fussy and can eat the HMO without a little bit of help. Luckily, other Bifs, such as B. bifidum, only want to access the juicier inner parts of the HMO, and while doing so, share their leftovers with M16V so that everybody's left feeling satisfied. So what about the next steps and what we've concluded from this work? First, it is important to begin expanding these HMO Biotic gene trait matching protocols established here to include a greater repertoire of probiotic species. Overall, we conclude that genotype, but also phenotype of strains should be considered when selecting a strain for probiotic use, as variations in HMO utilization may determine their fitness and their performance in the gut. Incorporation of additional complexity to mimic more natural settings will further our understanding of the functional implications of genotypic variations. And in summary, we demonstrate the benefits for the use of multi-strain bifidobacterial probiotics, since cooperation between strains may expand their HMO acquisition capabilities and help shift bacterial communities closer to those observed in breastfed infants. I started with H&H Group in 2017 as part of my master's internship, and I never left. At the time, it was known as Bias Time, and since that time, I have seen the group expand both within Ireland and globally throughout my PhD, and I am now grateful to have the opportunity to continue this journey with H&H as a full-time researcher. I begin this role at the end of January, and I look forward to continuing to enhance science-based knowledge of human nutrition. This area of research that I have been involved in and that I will continue to be involved in the future is an ever-changing landscape, which requires continuous innovation and engagement with this research. I aspire in the future for my research to have societal impact, and I believe that the work that I will be doing is meaningful. I look forward to being continuously challenged and stimulated, and look forward to continuing to meet and grow and learn from everybody involved in it. And finally, a few acknowledgements. I want to thank the entire team at the H&H Global Research and Technology Centre Ireland, but particularly Dr. Jonathan Lane. I want to thank the glycobiology team based in Teagasc Food Research Centre in Moore Park, led by Dr. Rita Hickey. I want to thank Professor Dow van Sinderen at ABC Microbiome Ireland and Dr. Rebecca Owens at Maynooth University Ireland.